Okay, good afternoon, students. Uh, it has been a long time that uh, almost a month back I have taken your classes up to the experimental designing, and now I am taking your next topic of introduction to population genetics. So, because today I am finding some difficulty in my uh, operating system, and I want to know that whether you people are able to see me in the video, because in my preview it is showing null. So if you are able to see me and the video is okay, we can go ahead. So just write in this chat box if you are able to see me, whether you are able to see and listen me. So please reply to this question. So I may continue. Okay, so Viplav has said that he is able to see. Shreya Mishra is also there. Nurag Patel. Okay. Okay, so it means we are okay with our setup, and we can start our next level of this genetics, that is the population genetics. So this part we can only start if you have the basic understanding of the statistics as well as those of the Mendelian genetics and the deviations from Mendelian genetics and mutation. Okay, so once you know this thing, so we can go ahead with the introduction to population genetics. And this particular topic, when we are starting, I have kept this lecture for introducing you to this particular subject, because when you were studying and preparing for medical examination, you might have come across many of the examples and things about the Mendelian genetics and its deviation. But very few people give emphasis on the population genetics. However, many a times there is a question in the need from the population genetics. So we will be looking more in detail about the intricacies of population genetics, and it is nothing but simply extending our knowledge, whatever we have gathered up till now, that of the statistics and that of the Mendelian genetics, to extend this knowledge on the population to understand the population structure and how the gene and the genetic composition of a population behaves over the time. Okay, so let us start with the introduction. And this introduction says like this, that it has remained one of the intrinsic question to mankind about how various species on this earth has evolved to its present form. So basically, we are seeing that we are surrounded by many species. Okay, human are there, then other species, canines, felines, and all these species are there. So how these all these life forms have evolved? Okay, this is one of the basic intrinsic questions that remain there with the mankind. Okay, so the population of a species have traversed through various variation and keep on changing with time. So what from our knowledge, scientific knowledge we have gathered, we understood that species what, say for example, Homo sapiens. Okay, so it is not like that these Homo sapiens were same as we are seeing today. At one point of time, they have traversed, this species have traversed through various variation, and it keep on changing with time. Okay, now, to understand these things, it becomes foremost to study what constitutes a population, its genetic structure, and how it get changes over time. So these are our basic central idea that what constitute a population. Okay, that is the one thing. Then what is its genetic structure, right? And then how it gets changed over the time. So these are some of the basic questions we will be looking into the population genetics to answer them and to quantify them. Not only just to answer them, we will also try to quantify them. That is how we can calculate these values. Okay, so Now, the population genetics is a subfield of genetics. Okay, of course, population genetics is a subfield of genetics that tries to answer these questions. Okay, the questions which I have asked earlier in the previous lines. So, now the population genetics have been defined in various ways by different people as given below. So, we can see that many people have defined this branch of genetics in different ways. So what this American Heritage Stedman's Medical Dictionary says? So it says that the population genetics is the branch of genetics that deals with the genetic makeup of population. So it defines the population genetics as the branch of genetics that deals with the genetic makeup of population. Right? Now, similarly, American Heritage Science Dictionary defines it like this, that the scientific study of the inheritance and prevalence of genes in population. So up till now, what you were studying in the Mendelian genetics was that there is a father, there is a mother, okay, then they have a genes, we have a particular traits for there were genes were there, how they get united and produced in the progeny. But now we will be studying this and extend to the level of population instead of individuals. Okay, so what this particular definition says, this particular definition says the scientific study of inheritance and prevalence of genes in populations, usually using statistical analysis. So here it comes that statistical knowledge is necessary for any geneticist to understand the population genetics. Okay, if you do not know the statistical analysis, you will not be in a position to understand the intricacies and the numerical formulas which are used to estimate various constituent structures of a population. Those are involved in the flow of genes. Okay, so now next is the New Dictionary of Cultural Literacy, third edition. It has defined the population genetics like this: the study of the genetic composition of population in order to understand the evolutionary forces that select for a particular gene. So what they are saying is that we are going to study the genetic composition of population in order to understand the evolutionary forces. So every time there are some natural forces, natural selection is a ongoing process, and those evolutionary forces act on the genetic composition of the population for a particular gene or a set of genes and made them to change. And when they changes, the structure also changes and the appearance of the population also changes, and how it all occurs is known as population genetics. Now, Wikipedia defines this as the population genetics is a subfield of genetics that deals with the genetic differences within and between populations. So when we were studying the analysis of variance technique, we were having two groups, two treatments and two populations. So here also same thing. So what we are going to do in the population genetics is that we want to understand the genetic differences, not the phenotype. Okay, we want to understand the genetic differences within and between populations. Okay, and is a part of the evolutionary biology. So evolutionary biology itself is a subject, and it is the part, population genetics is the part of that subject, and it is a subfield of genetics that deals with the genetic differences within and between populations. Okay, now, there is another document in the Stanford Encyclopedia of Philosophy where Okasha Samir has defined population genetics like that population genetics is a field of biology that studies the genetic composition of biological population. So ultimately what you have to do? You have to study the genetic composition of biological population and the changes of genetic composition that results from the operation of various factors including natural selection. So here in this population genetics, what we actually understood is that this population genetics has something to deal with the genetic composition of biological population or any population, and there are some factors which are acting on it in order to change the genetic composition slowly and slowly over the time, and it also includes the natural selection process. And by understanding all these things, if this is the field, or we can say the subfield of genetics, which is also the part of the evolutionary biology. Okay, so different people have defined this population genetics in different ways, and my idea to give you these different ways is that by understanding these different ways, you will be able to understand the intricacies of the subject. Okay, because in definition there are hidden things, those explains that what you are going to study in future. Okay, so let us move on to the history, because in any topic history is very important. That tells us that our ancestors, what they have already done, what were their views, and how those views continuously changes over the time to give us the extract of that knowledge that constitute this population genetic subject. Okay, so I will be starting my this history from the Darwin period. Okay, so the history of population genetics can be traced back to the work of Darwin. So you all know this thing that Darwin has published in 1859 one of his remarkable collection of his reports in form of a book, and which he named as Origin of Species. Okay, so in that Darwin has proposed two theories basically. And what were those theories? That modern species were descendant from common ancestors. That whatever the modern species we are seeing today, they are actually a descendant from a common ancestor. Okay, there was initially a common ancestor, and there is bifurcation of the branches, and that is why we are able to see the species which are reproductively isolated. That is, they cannot uh, naturally reproduce between them. Okay, so this is the first theory this Darwin has proposed. Then, second theory Darwin has proposed was that the process of natural selection was the major mechanism of evolutionary change. Okay, so for him, he told that it is the natural selection which is actually causing the evolutionary changes, and we have descended from a common ancestor, and now you can see lot of variation between the modern species. Okay, so it is the second theory over which heated debate continued for decades before geneticists could come to a common consensus. Okay, second theory, on the second theory there was a heated debate that how this process of natural selection can be claimed as a major mechanism of evolutionary change. Okay, so basically Fleming Jenkins, he was one of the scientists. Okay, and what he famously objected to Darwin theory is that the natural selection to gradually modify a population over a long period of time in the manner suggested by Darwin, a continual supply of variation is needed. That is, if we want to make the new species from a common ancestor in such a long period of time, continuous supply of variation is needed. Okay, then Jenkins argued that giving blending inheritance, before that period it was the basic concept of blending inheritance was there. Okay, that is, Jenkins argued that the blending inheritance, a sexually reproducing population will become phenotypically homogeneous in few generations. Okay, that is, if there, even if there is a change, and you do not have the variation, needed variation within the population, how the progeny will change from its population. Okay, for the progeny to be different from a population, it is required that the trait we are studying, it should be variable in the population. If all the people are of same height, the progeny will be of same height. You can increase or decrease the population height only if there is a variation in the height of the parent population. Okay, And if it is the blending inheritance, that is there is a admixture of the things, so that a sexually reproducing population will become phenotypically homogeneous, far shorter than the number of generation needed for natural selection to produce complex adaptations. Okay, and Darwin was not able to provide a satisfactory answer to the objection against his theory, that is the theory of the Jenkins, that if we are to consider the blending inheritance, how you can supply a continuous variation, because that get abolished within short number of generations. So this is because the scientific community at that time was not having a proper understanding of inheritance mechanism. Okay, that is people were not knowing that how the inheritance mechanism was there. Okay, now it is the Gregor Mendel in 1865 and 1866 proposed the principles of inheritance. Okay, you know these things, that Gregor John Mendel is the father of genetics, proposed the principles of inheritance. Okay, obviously our modern understanding of heredity is vastly more sophisticated than Mendel's, but key elements of Mendel theory, that is discrete hereditary particles, the genes. He called these as discrete hereditary particles that come in different types, dominance and recessiveness, and the law of segregation have turned out to be essentially correct. Whatever now our knowledge has become very sophisticated, but if we consider a one gene at a time and consider those discrete hereditary particles, then these dominance, recessiveness, law of segregation, everything comes out to be essentially correct. And that is why the Mendelian, these factors and genes of modern population genetics. So these Mendel factors are the genes of modern population genetics. And the alternative forms of that factor can take, that is, say, for example, round versus wrinkled in pea plant, are known as alleles of a gene. So what Mendel has told as factors are the our genes of the modern population genetics, and the alternative forms of that factor are known as alleles. Right? Now, the law of segregation is explained by the fact that during gametogenesis, each gamete sac cells receive only one of each chromosome pair from its parent organism. Now, other aspects of Mendel theory have been modified in the light of later discoveries. So, whatever aspects Mendel has given, many of them have been modified. Say, for example, linkage is there, epistasis is there. Okay. Now, Mendel thought that most phenotypic traits were controlled by a single pair of factors. Okay. This line is very important, that Mendel thought that most phenotypic traits were controlled by a single pair of factors like seed shape in his pea plant. But it is now known that most of the traits are affected by many pair of genes. Okay, it is now known that most of the traits, if you take most of the traits, they are not affected by single pair of genes. Rather, they are being affected by many pair of genes and not just the one. Right? Now, the Mendel believed that the pair of factors responsible for different traits, that is seed shape or flower color, segregate independently of each other. Okay, that was his next thing now. Okay, that these things get segregated independently, but now we know that this need not to be so due to linkage. That is due to linkage, they, there may be chances that they may not segregate independently. Despite these points, Mendel's theory marks a turning point in understanding in our understanding about the inheritance. Okay, so it is in the year 1900 when the rediscovery of Mendel work has taken place. And the rediscovery of Mendel work in 1900 did not lead the scientific community to be converted to Mendelism overnight. Because you all know that Mendel has although discovered these things way back, but it is only the rediscovery of the Mendel work that has brought this to the scientific community. Okay, and it is not like that within few days these everybody start thinking that okay, it is only the Mendel laws which are applied. Okay, now the dominant approach to study of heredity at that time was actually biometry. Okay, and it was spearheaded by the Carl Pearson in London. It is the same person who has discovered Carl Pearson coefficient of correlation. Okay, so at that time during the 1900, people were more interested in the hereditary mechanism involving the statistical methods and biometry, which involve the statistical analysis of phenotypic variation found in the natural populations. Okay, so they were engaged in the analysis of phenotypic variation found in the natural population. So why I am making you study all these things is so that you can make a connect between the classical genetics and the population genetics. That is how these things are get interconnected. Okay, so that is why we are studying these history points in detail. Now, biometricians were mainly interested in continuously varying traits. So it is very important because these are the traits which are of economic importance. So these traits like height rather than discrete traits such as seed shape uh, that Mendel studied and were generally believers in Darwinian gradualism. So these biometricians were the believers in the Darwinian gradualism rather than the sudden changes that has been proposed by the Mendel. Okay, so opposed to the biometricians were the Mendelians, spearheaded by William Batson. So he is the another scientist who has in the favor of this Mendel discoveries and who emphasized that discontinuous variation and believed that the major adaptive changes could be produced by single mutational steps. So what he said is that major adaptive changes in a species can be produced by a single mutational step. That is, if there is a mutation at a particular one gene, that can lead to major adaptive changes in that population rather than by a cumulative natural selection as described by the Darwin. So a heated controversy between the biometricians and Mendelians ensued. Okay, many people have put forward their theories, their experiments and other things to prove themselves. As a result, Mendelian inheritance came to be associated with an anti-Darwinian view of evolution in the history. So what we are studying this Mendelian inheritance that had been associated as anti-Darwinian view of evolution. Okay, now it is in the year 1908 the Hardy-Weinberg principle was discovered by G.H. Hardy and W. Weinberg. Okay, so these were the two independent scientists who have discovered the Hardy-Weinberg principle in the year 2000, in year 1908 and is one of the simplest and most important principles in population genetics. Okay, so I will be showing you this in the third lecture that how these are derived and how it is very important. Okay, so the importance of Hardy-Weinberg principle lies in the fact that it contains the solution to the problem of blending that troubled Darwin. As we saw, that the Jenkins argued that with sexual reproduction, the variation in the population would be exhausted very rapidly. But Hardy-Weinberg principle teaches us that this is not so. Another benefit of Hardy-Weinberg principle is that it greatly simplifies the task of modeling evolutionary changes. That is, now we are able to compute and model these evolutionary changes using this Hardy-Weinberg law, and it has proved that it the variation we get in a population does not simply get dissolved or get ended within few generations. Okay, so because it is not the blending inheritance that can take place, instead that will flow from one generation to another. Okay, so when a population is in Hardy-Weinberg equilibrium, it is possible to track the genotypic composition of population by directly tracking the allelic frequencies. Okay, so if you know the derivation which has been given by the Hardy-Weinberg, so you will be able to, it is possible to track the genotypic composition of the population by directly tracking the allelic frequencies. Okay, now population genetics as we know it today arose from the need to reconcile Mendel with Darwin. It is not until unless that we reconcile the concept of Mendel and that of Darwin, our knowledge has finally get polished, that how this all is happening. So a need which become interestingly urgent as the empirical evidence for Mendelian inheritance began to pile up. Okay, because people started doing experiments and they were proving that if we take into consideration a single gene which are affecting a single trait, we are getting these results. Okay, now the biometricians, the first significant milestone was the R.A. Fisher's 1918 paper. Okay, and that paper was the correlation between relatives on the supposition of Mendelian inheritance. Okay, which showed how the biometrical and Mendelian research tradition could be unified into one. And Fisher demonstrated that if a given continuous trait, for example height, was affected by large number of Mendelian factors, do not consider them as a discrete. Do not consider that one gene is affecting the height. Okay, many genes are affecting the height, and all those genes are actually the large number of Mendelian genes, each of which made a small difference to the trait. Okay, this is very important, that what he has proposed is that if given continuous trait height was affected by large number of Mendelian factor, okay, each of which made a difference to that trait, a small difference to that trait. Okay, this is very important. Okay, then the trait would show approximately normal distribution in the population. So this was the discovered by R. Fisher, that you take many genes which are actually affecting a continuous trait, and you consider that each gene is contributing a small contribution into the trait, then what you will see that in a population, because of the different combinations of those genes, the height will assume a normally distribution. So now let us see this thing. Say for example, I am just making an hypothesis that height is affected by 25 genes. Okay, so we will be having so many genotypes. Okay, that we will cover up in the coming lectures, that how many genotypes will be there. Okay, and when these genotypes will be there, it is the probability of getting a particular combination in an individual is also different in the population. And when you will combine them, so what you will see, that if you consider that this gene number one is contributing say for example this much of height, a small fraction 0.002, then second gene is contributing 0.004, then third is contributing 0.006, then fourth is contributing 0.001. Okay, and if we will find that which combination is available, okay, and if I plot according to those combinations, so what you will find that the height as a resultant of the combination of these genes will follow a normal distribution. This is what was suggested by R.A. Fisher. Okay, so simply he told that you consider that many genes are contributing to a quantitative character. Each gene is contributing a small fraction to that trait. The combinations available in the population will definitely contribute that trait in such a way that it will follow a normal distribution. Okay, so he was the first to unite the concept of the Mendelian genetics to that of the Darwinian genetics. So, since the Darwinian process was widely believed to work best on the continuously varying traits, showing that the distribution of such a trait was compatible with Mendelism was an important step towards reconciling Darwin with Mendel. Okay, so I am stopping this session here. So, we will again get connected to you people at after say 15 minutes. So the next class, the next session of this topic will start at 4 o'clock. So since then, you can take rest, you can have your water, and if some snacks you want, okay. So we will again meet. Now we have gone up to the point in the history where we have reconciled the Darwin with the Mendel, and how biostatisticians have found ultimately the underlying principle that how a Mendelian genetics can be applied over a population. Okay.